across the world, and some of you are from Tamil Nadu, some of you are from Russia and Japan and Germany. So I'm glad to hear and I'm glad to see that you are watching my videos, and best of luck for your future endeavors. Here, DNA is a genetic material from the experiment of Alfred Hershey and Martha Chase in 1952. They worked on a virus that can infect the bacteria called bacteriophage. The bacteriophage is a virus, actually it is a virus that can infect the bacteria, hence it is called bacteriophage. Bacteriophages attach to the bacteria, and it is genetic material, then enters the bacterial cell. If you take the bacterial cell, here the bacterial cell can treat viral genetic material as its was own genetic material. In the subsequent manufacturing times or multiple manufacturing times, virus particles whatever present in it, these virus particles will be produced in multiple number. Hershey and Chase worked to discover it was a protein or DNA that virus entered the bacteria. They grow some viruses on a medium. So what they did, so they grew some of the viruses on a medium that contained radioactive, what is that? Radioactive phosphorus. Okay, and some other viruses on the medium that contain radioactive sulfur. Okay, viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not a radioactive protein. So here DNA contains phosphorus but protein does not, isn't it? Here similarly, if you take the viruses grown on the radioactive sulfur, contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur, it contains phosphoric acid. Bacteriophages or radioactive phages, when they allowed to attach to the E. coli bacteria, okay, let us take the radioactive phases. Okay, these radioactive phases, if they were allowed to attach to the E. coli bacteria, then as the infection proceeded, the viral coats were removed. Okay, the viral coats were removed from the bacteria. Hesitating them in a blender. Now, the virus particles were separated. If you take the virus particles, they are completely separated. After separation of virus particles from the bacteria, okay, by spinning them in a centrifugal machine, okay, if they spin in a centrifugal machine, bacteria which was infected with virus that had a radioactive DNA, were radioactive, indicating that DNA was the genetic material that passed from the virus to bacteria. Simultaneously, it is correct. Bacteria that were infected with virus that had radioactive proteins were not radioactive, but this indicates that if you take the proteins, here this indicates that proteins did not enter the bacteria from the virus. Here, why? Because proteins are macromolecules and they can't pass. DNA is therefore as a genetic material. Can be considered. Here, this is the experimental setup taken by the Hershey and Martha. This is a bacteriophage 1 and bacteriophage 2. Bacteriophage 1 contained radioactive sulfur labeled protein capsule, and bacteriophage 2 radioactive phosphorus labeled DNA. Okay. Now if they infect the E. coli bacteria, imagine this is a E. coli bacteria. Okay. In this diagram you can clearly see here both the bacteriophages, if they infect to the bacterial cell. Okay. Finally, no radioactive sulfur detected in the cells. In the cells of E. coli, no radioactive sulfur detected in the cells. Why? Because radioactive sulfur detected in the supernants or the supernants of the bacteriophages. Here supernants are nothing but bacteriophages. What are they? Initial bacteriophages are supernants. Okay. Why? Because when the bacteriophages releases their genetic material into the bacterial cell, okay, they undergoes for further changes with the bacterial cell genetic material. Whereas here also phosphorus labeled DNA, radioactive DNA, when these phosphorus labeled bacteriophage infect the E. coli cells, what happened? Finally, blending occurred, and finally after centrifugation and in the centrifugal machine, when they spinned, what happened? Radioactive phosphor detected in the cells inside the bacterial cells, but no radioactive detected in the supernants of the bacteriophages. Okay, it is for negative and it is for positive. Understood everyone? This is the experimental setup done by the Hershey and Chase experiment to prove that DNA is a genetic material. Understood everyone? Remaining part will continue in the next video. Thank you.